hope I didn't get you all up too early this morning. So,
Colin, we're surgeons. We're up all the time at this time.
It was a joke, Martin.
Okay.
So the question really is, what can be learned from a single cell? And I think to begin answering this, it's worth remembering that I run a genomics core at the Vancouver Prostate Center, which always serves as an innovation incubator, developing new technologies and tools, both physical tools and computational tools, that are then made available to the Prostate Center and UBC community and beyond. And so really our tools range from everything from DNA sequencers, single cell library instruments to fairly sophisticated computer science and machine learning for predicting what drug might work with what patient. And recently we acquired a new DNA sequencer from BGI and their partner MGI. And the advantage of this sequencer is that it's very fast turnaround and very inexpensive. So while it can't compete with the real big boy sequencers in terms of cost, it has the advantage that if you want to try things quickly, you don't have to ship off your sequencing libraries and then wait a month. It would be on the order of a week, and the cost would be extremely favorable for local researchers. So about a year and a half ago, Stefan, I'm sure all of you know, convinced me that we really needed to get into single cell sequencing. And we had money from a CFI grant, so I acquiesced. And I thought I would share with you what we've been learning on this very interesting road to learning about single cell sequencing. So, with bulk sequencing, which is normally what most people still do, you might take your tumor, grind it up and sequence it, see what the average clone looks like, but never the rare clones, and then match that to a known drug or do your biology. With single cell analysis, you dissociate the tumor. You don't grind it up. You dissociate it into individual clones, and then you sequence each one of these. We're doing RNA-seq, so we're getting gene expression in say 5,000 to 10,000 individual cells that then allow precision drugs to be delivered to subclonal populations if you choose, or to learn about those subclonal populations. And this is great for things like longitudinal profiling. So monitor, intervene as the number of bad cells start to expand. And you can do pre and post treatment analysis with this. Not shown in this figure is the ability to characterize the dynamic ecosystem that exists between the tumor and its microenvironment. And I'll show data on that as we progress through the talk. So, what can the core do for you using this kind of technology, and what's it doing for us? So this is what we use. We use what's called single cell RNA sequencing from 10x Genomics. This, I think this box was like $200,000 and the size of a toaster. I was a little disappointed when I saw the footprint relative to the cost, but effectively, you dissociate the tumor, feed it to this thing, and in no time at all, you have a library that can be sequenced, and then every single RNA can be tracked back to an individual cell. And And so you get the readout of this is you get clusters by cell types based on gene expression. So each cluster will be defined by its repertoire of gene expression. It defines system heterogeneity, compares dynamic gene expression over time, and defines cell type of origin. And it's fairly straightforward to monitor things like adaptive responses and novel biology. So this, as you can imagine, this is fairly complicated computationally, and so this is sort of the pipeline of analysis that we use. Some of this is, you know, comes with a box, and others we've had to develop as we learn about the complexities of this kind of data. Sometimes you read about this single cell sequencing and it sounds relatively straightforward, but it turns out it's anything but that. And that there's an awful lot of work that has to go into the analysis of the data, or actually at all levels. But certainly the analysis is very tricky because there's all kinds of room for artifact to come into it, from making the libraries to getting the data to analyzing the data. So I like to think of this really as a virtuous circle where we start with clinical biological questions. That goes to computer science and statistics, and then back to more refined questions. And this requires people like Yenny Lin, who's a computer scientist and genome analysis expert, Stas Volek, who I always sort of refer to as a resident genius, who's a computational biologist, and Robert Bell, who most people think is a physicist, but in fact, he's a physicist who worked at CERN and is really an expert in thinking mathematically and using tools like R. So, it requires this kind of human ecosystem to work with this kind of data and to interact with local collaborators. So if we go back a number of years, YZ Wang developed a model for neuroendocrine cancer that I think remains the only one in the world that transdifferentiates from an adenocarcinoma to a neuroendocrine cancer. And this just shows that he put a patient tumor in one of his mice, let it grow up. It was expressing AR, PSA. He castrated, waited some amount of time. It turned into a neuroendocrine cancer, as shown by expression of synaptophysin, at which point he came running into my office with ideas on how to spend my money. And so, again, we were interested in dissecting this phase or state transition in the neuroendocrine. And, again, this is an incredibly powerful model because you can track it across time in a time series. So you can look before it transdifferentiates, while it's doing whatever it does, before it terminally differentiates into a neuroendocrine tumor. And this is just looking at various markers, for example, AR, which stays on for a very long time, even when PSA is switched off. And then we see Chromogranin come up at the very end. So this is what, and then this is the kind of analysis we could get the old way, looking at bulk sequence, and this is very powerful, right? We learned a lot. I think a number of the genes that are now known to be drivers of this neuroendocrine transdifferentiation came out of the Vancouver Prostate Center, have been validated in large cohorts, and are becoming therapeutic targets. So this is in fact what was in fact very powerful. But this is the old way. This is grind up the tumor, throw it into a sequencer, get bulk data, see the dominant players, and and make progress without a doubt. But this is the new way. So this is basically that same experiment. What I'm showing you here is pre-castrate adenocarcinoma. So this is grown up in the mouse. It's expressing AR and PSA. Castrate, and you get time point 1, time point 2, and then terminal differentiation to a neuroendocrine tumor. So what am I showing you here? So if we look in this quadrant here, we can see these different groups of cells based on color, and I've labeled them A star, A star star, B star, C, and D. So this is the transcriptional heterogeneity that exists in the pre-castrate tumor. And in this case, we see in this case, we see as few as D is as few as 2 cells. So we see 2 cells there. And I've labeled them according to whether AR expression is weak or absent or AR strong. So you're looking at heterogeneity not only in transcription, but at the level of AR. So then we castrate, and you can see that these 2 cells now enlarge. The scattered blue cells become more dominant. And where they were weak, now they're both strong and weak expressors of AR. The orange cells, may have decreased, but then the blue cells have expanded substantially. And these gray cells, which are strong expressors of AR, appear to have undergone an extinction event. They're just not there. And then at a later time point, these are individual time points from different animals. So this speaks to the reproducibility as well. You can see that the general pattern of yellow, blue, and orange cells is identical. But what we noticed was a small group of cells out here that are chromogranin positive. And so we refer to these as emergent neuroendocrine-like cells. I'm sorry, this is mislabeled. They are AR positive. And then they, at the next time point, we have full-blown neuroendocrine tumor, which interestingly, if we look at it carefully, is heterogeneous as well. So there's about 75% of the cells are classical AR negative, chromogranin positive, and about 25% are double negative, so AR negative, chromogranin negative. And then there's these other cells that are mixed in terms of AR and chromogranin, sort of green, bluish, and gray. And we're digging in to understand what this means now. But you can imagine that just this quadrant here poses all kinds of interesting questions. I mean, why are some of these cells resistant to castration? What are the underlying gene expression profiles? How do they change and adapt to the castrate environment? And then how do these emergent cells emerge, right? I mean, and so we have all the gene expression to dig down into that. So this is sort of single cell pathology, where we can just look and see what the staining, quote, of AR and chromogranin A looks like in these cell populations. And so again, we can see that AR is staining all of the cells here, some much more strongly and uniformly, others sort of mixed, and how that changes over time. And then as we enter neuroendocrine, it vanishes. And we can see in those emergent cells down here that it's negative that they're negative for AR. Likewise, here's chromogranin A, and we see it's negative, and then we see the emergent cells express it, and one cell here expresses it. So we might wonder if that this is the population of cells that are converting epigenetically or otherwise to this phenotype that we see here, and then we can see the very strong staining in the neuroendocrine state. But if you look carefully, you can see gray cells dotted throughout this, meaning they're negative for chromogranin A.
Colin, let me just stay on that slide for a second. What does the spatial localization represent in this?
So, for example, these here.
Yeah. Well, yeah, everywhere where they are.
So those are clusters of cells that have been assigned different space based on different gene expression. So their gene expression can be similar but not identical.
Just to clarify, though, that it does not represent where they sit within a tumor.
No. This is not spatial.
Right?
This is not spatial. This is just telling us that these are distinct populations of cells defined by gene expression.
Colin, this is Amina.
Yes, Amina.
Yes, when we're looking, we can definitely see here that certain population are not transdifferentiation, definitely. When you look for this population with IR negative, that you cannot talk about transdifferentiation. The only thing, especially those one that they are low on androgen receptor, because you just have enhancements. It's mean what you see here is not only a transdifferentiation. It is also a residing population that are expanded after hormone therapy. Is that right?
I mean, I think so.
Yeah. The other thing I would like
this remains this is all brand new data, and it's very much a work in progress.
The other thing I have to tell you in regarding to this data, I think that you are restricting yourself to chromogranin, but not necessarily that the only thing that you have to be looking at. When we look back to a lot of studies in single cells, especially in relationship to some emergent phenotype, you got to look more carefully about a signature rather than a marker that perhaps will enhance and straighten the data better than looking specifically to chromogranin, which not necessarily is seen also in all the patients that they have neuroendocrine prostate cancer. I believe that's what you have here, both expanded pre-existing population and somehow some transdifferentiation, but it's better to look at it at the signature point of view to have more powerful data, I think.
Yeah, as I said, this is a very much a work in progress, and to be honest, the where we've spent most of our time is getting rid of all the artifact that we're. Luckily, this comes from a hybrid system, so there's mouse genes here as well. So we were able to identify the artifact. I think there's going to be a lot of retractions in the literature over that issue, although people are now recognizing it. So, then we went on.
The other thing I just want to say, because it's a transdifferentiation model that you're working on, and you have just to be careful when you put in your Disney plot and analyzing them, be careful about also looking to do them with the cell cycle, because that can affect also the population when you're analyzing them.
Correct.
Okay. That's it. Just gonna give a need, and perhaps you will see more emergent population than rather just this chromogranin, because with castration you're gonna have shrinkage, a lot of effects on the cell cycle.
Correct. So, but at this stage, we basically just wanted to look and see, do we see what we think we should see at all? Right. That this was this is for new data. This is our older data. Just looking to see, do we see what we expect to see from the literature? And here we were just looking to see if the neuroendocrine cells are retaining an epithelial-like phenotype as reported, and we see that they do. Again, there's some heterogeneity in expression, which is expected. Then we looked at things like one cut and PEG-10, and so things seem okay at this stage. So then we want to look at sort of all known neuroendocrine markers, or not all of them, but but a fairly large repertoire of them to look across this time series to see how gene expression is changing. And again, this is relatively new data, and it's a headache keeping all these cluster names straight and with different people working with the data. But so we can look over here and we can see the androgen receptor expression, which drops off as we head into the neuroendocrine state, some of its target genes dropping off. And then we can see a whole range of neuroendocrine genes that are expressed pretty much as we would expect. And I've labeled those emergent cells, or whatever they are, those 5 cells here. So we can see that the brown bars represent gene expression switching on in those 5 cells. Things like PEG10, SOX2, Chromogranin A, ASCL1, SRM4, so on and so forth. So, we continue to dig into this data to make sure that what we're seeing is what we should see. And again, in the neuroendocrine, you can see, I think SOX2 is a good example, where there's 3 populations in that tumor, and and those are defined by this big blue bar, the green bar, and this lower bar back here. This gets back to the point Amina made, that you can't just look at, for example, Chromogranin A. We want to look at as many genes as we can to understand what the situation is and what we're dealing with. So then we can dig in deeper. We can say, okay, well, within that neuroendocrine tumor, again, you can see these are those emergent clones here, and then the neuroendocrine. We thought, well, that sort of looks like 2 different clusters, and so we analyzed the gene expression between those 2 clusters, which are both positive. The bulk of them are positive for Chromogranin A, and we see that the lower one, circled in red, is much more active, it looks like, proliferatively than this one. There's more work to do on that, obviously, but we see cell cycle. When we do gene set enrichment analysis, we see cell cycle mitotic M phase, on and on and on. But that's not so interesting. That's neuroendocrine. It's cool that there's heterogeneity, but this is the analysis that looked more interesting to me. And this is when we look at those emergent cells relative to the neuroendocrine. And we look at, genes that are 4-fold different, 4-fold higher here than here. And then we do gene set enrichment, and suddenly we're finding things, developmental processes, extracellular matrix, collagens, on and on and on. And when we dig into the genes involved, there are things like 6-3 shown here, which is a gene involved in the development of the eye, central nervous system, and in fact the brain. And so there's quite a few genes involved, expressed in this stage, that look to be involved in neurogenesis, to use one word. And so then we were able to dig in more deeply into those 5 cells. And again, I'm not saying any of this is like 100%, because we're still digging and validating and trying to understand the data. But it is extremely interesting that a number of the genes we find are involved in neurogenesis, and that's relative to the neuroendocrine state. So again, we see 6-3. These are these arrows indicate sort of controls AR expression, chromogranin A, PSA, and REST. So we see genes like HES6, they're relatively unknown, DLK1, so on and so forth. And these annotation, this would mean this gene comes up in that early stage and then goes away or is decreased later on. Once you have transdifferentiation, it comes up in that intermediate or 5-cell stage and stays on. But there's heterogeneity of expression, obviously. And then the circles mean that they're transcription factors or they're involved in chromatin remodeling. So this is just another way to look at that Robert came up with. That's interesting, but is less interesting. So,
Colin, if you look globally to your gene set enrichment analysis in these 5 cells that you're mentioning here, you showed us specific genes, however, and they are involved in neurogenesis, most of them, and I'm aware, I know those genes pretty well, but if you look to different pathway that they are enriched or comparing them to other cells of population, what are the pathway that they are enriched, what are the pathway they are inhibited in this one? Did you do that, or you focus specifically to certain gene?
No, pathway analysis is coming.
Okay.
Again, we spent an inordinate amount of time getting rid of the artifact in this data and understanding where it's coming from and on and on and on. So, right now we are going from, gene set enrichment to pathway analysis. But we're trying to do it systemically, and I'll show you what I mean in a minute. So yeah, there's a lot to do. I'm just trying to give an overview of what's possible here, and again, this is all pretty much brand new data. So one of the things that we can do easily, because this is a human tumor growing in a mouse, is that we can analyze the microenvironment. So if you imagine this is the patient-derived tumor growing in the mouse, it turns out that the microenvironment, the microenvironment or stroma gets replaced by mouse cells. And then, so all these fibroblasts and macrophages and so on and so forth will be replaced by mouse cells. And then the tumor will reprogram them to be human-like. And this is all data that's published supporting that. And there's lots of evidence now that, the human stroma gets replaced by mouse, and then the mirroring analogs are reprogrammed by the tumor to be human-like. And so a while ago, postdoc in my lab, who's now back in China, Fan Mo, figured out how to do this computationally, how to separate the mouse genes from the human genes. And then we were able to show that, they retain a high degree of similarity with what was human, because they can be predictive of metastasis in very large data sets. And so I got at this from starting with mouse stromal cells, basically, and my good friend Massimo Loda got at it by microdissecting the stroma. So in fact, the mouse stroma in these PDXs does retain many of the characteristics of the human population. So we're able to, and this is much less developed data. But what I'm showing you here are clusters of cells now at each time point, pre-castration, post-castration, and neuroendocrine. And these are all fibroblast macrophages, endothelial cells, some kidney cells these are kidney cells here that were cut when the tumor was removed. adipocytes, and unknown clusters that we think are atypical fibroblasts. Again, this is all defined based on gene expression. And again, so macrophages are this color here, different population of macrophages shown here, so on and so forth. And again, we can go through these individual cells and time points, and now we're looking at, in the back, the gray bars are changes in gene expression in those 5 cells, or around those 5 cells. It's not in those cells; it's the stroma surrounding those 5 cells, and that sort of time point. So yeah, I mean, this is very interesting. What does it mean? So we reached out to my former PhD student, Ronick, and who's working with Felix Feng now at UCSF, and they've started looking at these genes in different neuroendocrine populations. And so I'm just showing you these here. So these are controls in this set of genes. These are some of our genes from the tumor, and then from the microenvironment here. Now, the challenge here is we're often looking at genes that are expressed in a small number of cells. So it is true that we will miss a lot, right? So if we're looking at low gene expression, and so, for example, a population of 10 or 20 cells, or 5 cells, or 1 cell, we probably won't see them in bulk sequence. But nonetheless, we can see, for example, in Beltran's data, that we're validating quite a number of interesting, or potentially very interesting genes. And again, this is brand new data. And so far, we've pulled out these genes: CHD7, HES6, MEOT, which is a long non-coding gene, PCP4, that validate in every cohort shown here. And then there's others that are linked to neuronal and CNS development that are expressed at low level in a few cells, and so aren't seen here, but are still going to be followed up. Now, one of the things we're interested in is understanding, how do the cells communicate, right? How does the tumor microenvironment communicate? And this is a paper published back in 2014, a draft network of ligand-receptor mediated multicellular signaling. And now we're working with Cenk Salhanap, who's at the National Cancer Institute now, who's doing similar work. But this is work just again, brand new. This could be completely wrong, but this is our first go at it. And so this is revealing communication networks between the 331 tumor and surrounding tumor microenvironment genes. And so we have ligands being secreted by the tumor, and then we have receptors shown on the different cell types. So this obviously requires a lot of validation, but on the other hand, it's a hell of a starting point for hypothesis formation and for looking at potential targeted therapies or repurposing existing therapies. So another question we're asking, again, this is brand new, is can metabolic vulnerabilities be leveraged for therapy? And so this is an ongoing collaboration with Xingjin Kung in Taipei and Paul Sorensen, who most of you know, I would imagine. And this is this started with my work in dormancy that has shown that proline metabolism appears to be important. And work from Singen that's shown that this gene ASS1 is a vulnerable point in tumors for therapy. Because arginine gets shifted to nucleotide synthesis, this gene ASS1 gets downregulated. That makes the tumors arginine oxytrophs. And that means if you either inhibit ASS1 or starve them for arginine, they die. And so looking through our data, we observed that the Prod H is in yellow here, is never really expressed. But consistent with previous data, ASS1 is expressed at low levels and then drops off in the neuroendocrine state. And what I'm showing you here is data from the lab in Taipei, showing that when you grow H660, which is a prostate cancer cell line, maybe neuroendocrine a bit like, when you grow it in arginine-restricted environment, it really doesn't grow. It just sits there versus control and versus 2 inhibitors of ASS1. This is more data showing the same thing, basically different prostate cancer cell lines and controls showing ASS1 mRNA, and then targeting it with an inhibitor, ADI1, or ADI rather, and just showing that the PC3 and H660 are extremely affected. CWR is affected, but the control grows fine. And interestingly, in metabolomics shows a drop in arginine and proline metabolism, meaning they may be linked. Now, the interesting thing about this is that when you starve them for arginine or you inhibit ASS1, you kill the mitochondria, and then you start getting leaking of DNA from the nucleus, and that triggers the STING pathway, so that you also get an immune response, if I have that correct. So you both are killing the tumor cells and bringing in the immune system, and this is just data showing that this works just fine in xenografts, the arginine-free diet. So I'm gonna end now with, this is a study from Shanghai that was published in bioRxiv, and this is basically translating single cell sequencing to the clinic, where you go from a biopsy, enzymatic digestion, to sequencing, and they were able to look at neuroendocrine transdifferentiation occurring in patient tumors using this technique. This is a reasonably interesting paper, and I would encourage people who are interested to look at it. And this is what we're trying to do at the Prostate Center now. So it turns out that fresh biopsies are fine. You can dissociate and sequence those. But if they're frozen or they're in OCT, you can't do single cell sequencing because the cells will have lysed. And even if you use cryopreservation, there'll be a lot of dead cells. So, and that makes it messy. So what we're doing is single nuclei sequencing. And so this shows you where we're able to get single nuclei now from OCT. And then we do the sequencing. And this just shows that when we do this, the RNA tends to be okay. Again, this is somewhat old data. This is before COVID, and we're back at this now. Again, the advantage we have now is that we can probably by next week use the new DNA sequencer to do these at least 1/3 the cost. So we're no longer burning huge amounts of money for R&D. But this is coming along, and this will work with biopsies. And we're working with various people around North America on this. So with that, I will end and say thank you and take any questions.